Dear students, welcome and thank you for tuning in for this video. We are starting a new lecture in the Biomaterials class, which is related to the structural material of the body and protein synthesis. This topic is very important. We need to understand how proteins are made and what kind of structural materials are found in the body, that they're made actually mainly of proteins, because we would like to understand the basic structure of proteins and how they're made, the process in which they're made along the way, so that when we want to intervene in this process or understand the process more, we need to have the basic knowledge of protein synthesis and all the structural material inside the body. The question that we want to ask and answer is actually, how are the structural materials of the body are made? What are the steps? What are the components that they play a critical role in this process? So if you remember from the previous videos, we talked about categories of organic biomolecules, the biomolecules inside the body. And in Biomaterials 1, we are focusing on understanding the interaction of the proteins and the biomolecules with whatever material we are implanting inside the body. And mainly the materials that we are implanting at this stage will be inorganic, metals, ceramics, and some of the other polymers, mainly ceramics and metals. So we talked about proteins, we talked about lipids, and we've described that there are functional and structural biomolecules. And also carbohydrates, as well as they are also could be functional and structural. But however, there is one category of organic biomolecules, which is the nucleic acids, which are only functional, they're not structural. So they don't support loads or withstand stresses and strains. So they're just only functional biomolecules. And we would like to understand the process of starting from nucleic acids, which are the DNA and the RNA, and then creating other functional and structural biomolecules such as proteins, lipids, and carbohydrates, and some carbohydrates. We would like to focus on protein synthesis, starting from the DNA until we have a functional protein. And this is the lecture that we're going to start in this video and the coming videos. We would like to understand the structural components inside the body, and we'd like to see how the protein synthesis as a process is developed and taking place in order to understand how can we intervene in the right way once we would want to do this. Let's start with the DNA and RNA. So those are nucleic acids, and we remember that we have 5 nitrogenous bases plus a ribose plus a phosphorus to create our nucleic acids. And adenine, guanine, cytosine, and thymine and uracil. Those are the nitrogenous bases. If we took a look at this picture, this is a single. If you can look at these stranded that's coming out of this cell. So this is a single DNA actually molecule spelled out from a ruptured bacterium cell. And you can see that this is just like a line, and it's a very long line. And this is similar for all cells. And this is a bacterial cell. If we would like to look at the human cells, so the DNA is almost 3 meters in length, which is approximately 10 feet. And inside our nucleus, we have the human genome is defined by about 23 pairs of chromosomes. And these 23 pairs of chromosomes, they hold 3.2 billion nucleotides. And when we say nucleotides, adenine, guanine, so these are the bases. So we have almost 3.2 billion of those in 23 pairs of chromosomes, and the total of 46 chromosomes. 22 of those pairs called autosomes, and they look the same in both males and females. The 23rd pair is the 6th chromosome, and they differ in males and females. Our genetic material, it consists of the DNA. We need to highlight here that the genome includes both the genes and non-coding DNA. And we need to understand the difference between the two. So in our 46 chromosomes, we have a clear set or sequence of nucleotide that they are identified as genes. So if we have, let's say, 3.2 billion nucleotide, we can find that there is, let's say, for a specific protein, let's say for example insulin, we have a number of nucleotides that they are coming in sequence in one place in one of the chromosomes. This sequence is the only sequence that can make insulin as a functional protein. That is a gene. That's what we call a gene. Now, we can find another gene that makes another hormone away from, the insulin gene, but between the two genes, so another, let's say collagen, so we can create collagen, and another gene, but between the two genes there is a spacing. So we have these nucleotides that they're not part of genes, so they don't be expressed or translated into proteins. We call these genes as non-coding DNA. And from a scientific point of view, it's important that we know we have our genes, that they can create for us functional proteins. Those spacings or non-coding DNA, they're very important because they're not going to create proteins, but the spacing, the size, and the frequency of these spaces actually play. There's a lot of research that indicate that it plays a huge role in the likelihood and life of the organism. So we just want to keep those in mind because we will going to come back to them again when we study more biomaterials and biomaterials 2, and when we look into tissue engineering. But this is what we want to learn at this moment. So we have genes. They are sequences of nucleotides that they can create protein for us, translate into protein. We're going to talk about this and use through the RNA. And then we have the non-coding DNA. Remember that we have also if you remember from biology, we have something called haploid and diploid. So haploid cells are cells that have only one of each of the chromosomes, so it's not paired. And those are the sex cells, sperms and eggs. And if we have the merging of the eggs and the sperms in fertilization, then we will have something called dipole cells. So these cells have the pairs of chromosomes. So if you have these pairs, it means that they are, together. It means that it's something called somatic cell, which means it have been fertilized. But if it's just the sperm and the eggs, we call them haploid cells because they have only one of them chromosomes. This is an introduction of how we're going to start our discussion about protein synthesis and structural materials inside the biological organism. Thank you for watching. Until next time. Bye.